Okay. Um, just give me a minute. Yeah. So, good evening, everyone. Good evening, everyone. Welcome to week 3 for the live interaction session for week 3 of the Basics of Biology course by NPTEL. So, as you already might remember, we have previously looked at, in case it's not, yeah. So we have previously looked at other topics for week 1 and week 2. And this time we are starting off with week 3, where the major theme was centered around the basics of cells. So what we will cover today in the live interaction session is to look at the week 3 assignment that you've already submitted. We will look at some sample problems for week 4, and we will overall revise whatever topics have already been covered as part of week 3. And just as a reintroduction, my name is Ankita. I'm one of your TAs for this course, and this is our plan for today. So first, we will be looking at, I hope this is visible on slideshow. So we will first look at the assignment for week 3. So the question which was given in the assignment, the first question was about when an isolated plasmid is run on an agarose shell, what is the order of its appearance? So before we get into the details, let us just try to recall what exactly is the purpose of running any sort of DNA sample on an agarose shell. Now, agarose gel, as you obviously already know, is used for DNA samples, is used for nucleic acids particularly, while STS page is used for protein samples. So, because DNA, by its inherent nature, because of the phosphodiester backbone, it has a negative charge along the entire backbone. Therefore, by the principle of getting attracted towards by the basic idea that positive charges attract negative charges and vice versa, since these molecules, these nucleic acid molecules, in this case DNA, have a negative charge on top of them, therefore when you place it in an electric field, it will migrate towards the positively charged electrode, which in this case is the anode. So here you can see it in the diagram. It has the answer already given. I'm just trying to revise the basic concept behind it. So when you initially load a sample, loading is introducing or um, adding the sample to the agarose gel in this particular condition. Then upon adding the sample, we apply an electric current across the gel. There is a gel apparatus within which there is a electric field which is applied. There's a positive electrode, there's a negative electrode, and samples are allowed to migrate. So under these circumstances, what happens is that DNA would be able to migrate from the negatively charged electrode, in whose direction is usually the wells that are loading wells that are present, and it will migrate towards the positively charged electrode over a course of time. So this migration is otherwise a better terminology for it is the electrophoretic mobility. Mobility means movement, and electrophoretic mobility is the term used in this case because there is an electric current which is responsible for this movement. Now, upon applying such a current, majorly what happens is for any molecule to migrate under the or in the presence of an electric current, there are two factors which determine how its mobility will actually progress. Majorly two factors, apart from other secondary factors. The first one is the charge. The second one is its conformation or size. So in this case, because we are only talking about DNA, these molecules are obviously negatively charged, and therefore all of the molecules will have the migration of these molecules will not depend um, upon the charge factor. It's no longer becoming a determining factor in this case. However, the second factor, which is conformation, that is more relevant under these circumstances. So conformation or size. Conformation basically means the arrangement or the three-dimensional organization or three-dimensional structure of the molecule. Now, when we are talking about a plasmid that has been isolated, then during the process of isolation, there can be different conformations in which the plasmid can be present. In this case, as you can see in the diagram and as has already been mentioned in the question as well, plasmids majorly can have three types of conformations. One is its normal covalently closed circular DNA, which is its normal form. The second is the open circle. Open circle can otherwise be considered to be nicked, which means that because you can imagine that DNA, it's a plasmid DNA, it's a double-stranded structure. Therefore, if there is a cut in only one of the two DNA strands, then you don't expect the entire molecule to unfold, right? But the opposite situation, in which case both these strands have a cut, under those circumstances, a circular thing will get linearized, right? So that is exactly what happens here. There are different conformations based on the kind of nicks that are present. So in this case, even though the question is not, the diagram that I've shown on the right is not exactly covering all of the options present, but you'll get a basic idea. So when a plasmid is supercoiled, one we talked about it being a covalently closed circular DNA, which is its normal conformation. There would be an open circle form and there would be a supercoiled form. So the supercoiled form is usually the one that has, as you can tell from the name, supercoiled means it has the maximum, what should I say, maximum compaction or maximum packaging, giving it the smallest conformation or the most compact or tight conformation which it can potentially have. So its migration among these three, among the covalently closed circular DNA, open circle and supercoiled DNA, the supercoiled plasmids will always have the fastest rate of migration or the fastest electrophoretic mobility. However, if you look at the other two, among the open circle and the covalently closed circular DNA, the open circle conformation has a faster rate of migration. This is an observed pattern that is usually seen. Here also you can see the same thing, that the nicked circular or the nicked conformation of the plasmid has a faster migration compared to the CCC DNA. And apart from that, in this case, RNA, you do not normally expect RNA to be present. But the fact of the matter is that during isolation, there is possibility. It should not technically not happen. You should have pure isolated plasmids, but there is potential for some sort of RNA contamination as well, even genomic DNA contamination, even though most of the time that contamination is avoided because of some of the steps that are included in the isolation process. But despite that, there is still possibility of the same, and usually it's observed as shear DNA and the same shear genomic DNA. And mostly the same situation is also true for RNA as well, where you can get sheared RNA, as a result of which they are sheared means it's very tiny pieces of RNA, as a result of which their size is small and therefore they migrate the fastest through the gel. So the correct option for this question is that you will have the loading well at the top, then the covalently closed circular DNA, which is a normal plasmid, which has not undergone any sort of compaction. So that will be slower migrating among all the other options. Then comes open circle, then comes the supercoiled DNA, as we have talked about it being the most compactly constructed structure, and then comes the RNA at the last. So this is the order of migration that is usually seen. Next, the second question for this week's assignment, for week 3 assignment, was about where does transcription take place in an animal cell. So when we talk about so when you get a question like this, first thing you need to address is what type of cell or what type of host system are they asking about. In this case, it's an animal cell, which obviously is a eukaryotic cell. So the reason why this is relevant is that if you consider a prokaryotic cell, for example, a bacterial cell, in those kinds of cells or in those primitive host bodies, what exactly happens is that there is no clear distinction between the sites or the locations where transcription and translation take place. There is no spatial distinction in the location where these processes take place. However, in eukaryotic cells, these locations are very important and they're very clearly distinguished between the two. So here we see that if you look at the options, then since only transcription has been specifically referred to here, there's nucleolus, ribosomes, nucleus, and Golgi apparatus. The answer for this would be the nucleus. If you, in contrast, if you take a bacterial or a prokaryotic cell into consideration, then transcription and co-transcriptional translation occurs in the cytosolic region, in the liquid part of the cell, and therefore such a clear distinction is not observed under such circumstances. Now we come to the next question, which had asked about which portion. This is a fairly direct question which has been discussed in the lectures as well. We will do a recap of the same also in a while. So here the question was referring to which portion of the mitochondria contains the enzymes that are required to run Krebs cycle. So obviously, Krebs cycle is relevant with respect to the mitochondria, but then as we have previously we have looked at in the lectures, that mitochondria is not like an ill-defined structure. It has layers, it has specific compartments, which if you come to define, then it has an outer membrane, it has an inner, it's a double membrane structure, right? Both the mitochondria and the chloroplast. So it has an outer and inner membrane. The gap between the two, or the space between the two, is referred to as the intermembrane space. Then there is the matrix and the mitochondrial cristae, which are the infoldings or invaginations of the inner membrane. So these are the subcompartments within an organelle, which is in this case, of course, the mitochondria, right? So here specifically we have been asked that where are these enzymes that are actually required for Krebs cycle to function, where are these enzymes exactly present? So as you already might know that Krebs cycle or the TCA cycle, or that is the tricarboxylic acid cycle, that occurs due to the enzymes, or that cycle is executed or carried out by the enzymes that are present within the mitochondrial matrix. So not the outer membrane, not the intermembrane space, not the inner membrane, not the cristae, but in the matrix itself, these enzymes are present. So this is a quite a direct answer, and I'm hoping that most of you might have gotten this question right. Then comes the question about where is chlorophyll localized in the chloroplast. So even though this too is a direct question, like these are about the compartments, these questions, they refer to the compartments or subcompartments within which these very crucial processes they take place. So they are fairly direct questions that you have to address based on the lectures that have been discussed, and you have to have a complete and thorough knowledge of what has already been taught. But apart from that, here also you can see the structure of the chloroplast. So I hope this is visible. Yeah, so here we have a cross section of the chloroplast. So there you can see the outer membrane, the inner membrane, the thylakoid, in this case the stroma. The stroma has these stacks of thylakoids which are called as granum, individual granum, plural grana, and these stacks of thylakoids are all together, the entire stack is called as that granum, and these are connected by stroma lamellae. So the chloroplast as well is quite it's a well developed organelle, also having a number of subcompartments, so that too you can kind of relate to the structures that we saw in case of mitochondria as well. So here are the substructures which are present. So the question in this case, it was about where is the chlorophyll. Look, just give me a second, sorry. So where is chlorophyll localized in the chloroplast? So chlorophyll, the pigment chlorophyll, it's if you look at the options here too, we have the outer membrane, inner chloroplast membrane, intermembrane space, and the thylakoids. And obviously, because photosynthesis itself also occurs within the thylakoids of these chloroplasts, so by extension, obviously the pig, but also by direct information as well, the thylakoids would be the region where we can expect pigments like chlorophyll to be present, and which would be responsible for carrying out the entire process of photosynthesis within a plant cell. So that completes question number 4, and then we come to question 5. So in this question, we have been asked which of the following in the chloroplast is responsible for absorbing photons. So if you just look above, we have discussed about the chloroplast, its structure, its cross section has been shown. Now within each of these thylakoids and along the thylakoid membrane, actually a huge number of proteins are present that are all, most of them, they are very crucial players um, in fine-tuning the process of photosynthesis and effectively in the survival of the plant, because that's responsible for producing food. It's a crucial process that is necessary for plants to survive. So some of these proteins which we've already studied are the photosystems. They are the key proteins that are extremely important when it comes to the process of photosynthesis. So mainly there are two photosystems, and in this diagram here you can see the Z scheme, that is the process of Z cycle, or the Z scheme, that is actually the process via which the absorption of photons and the hydrolysis of water, or the breakdown of water or the lysis of water, is effectively helping in the generation of energy equivalents like ATP, NADPH in this case, which is what the process of photosynthesis is technically about. So the question here has only asked us that which of the following among all of these proteins, you can see here there are like within the PS2 there's P680, here's P700, we can see QA, QB, pheophytine, other pigments, cytochrome B6F complex, and many other proteins, but which of them are actually responsible for absorbing photons, which is like one of the first key steps that even in the Z cycle you can see that H nu here is indicative of energy, and energy here is absorbed in terms of photons or in the form of photons, which is light energy, right? Quanta or packets of light energy are photons. So that is being absorbed by PS2, which is photosystem 2, which is basically what answers this question. But if you look at the other options here as well, you'll see that there is a translocon on outer chloroplast membrane, or TOC, the photosystem 1, photosystem 2, or translocon on the inner chloroplast membrane, which is TIC. So among these, the correct option, as you can obviously see in the diagram as well, is photosystem 2, and that will cover the scope of this question. Next, here is a very relevant question if you are looking forward to, or if anybody is actually practicing mammalian cell culture in their laboratory or in some scope of their research, then you, under most circumstances, unless you're performing a very special experiment where an indicator would not be desired, under most circumstances, a pH indicator is always added in a medium. The reason behind this is twofold. A, because it gives you an idea of the metabolites that are being produced by the cells that are growing. Because obviously, when you just adding a set volume of media one fine day will not allow the cells to proliferate, grow, survive over an extended period of time, or will not allow them to survive indefinitely. That media needs to be replenished because that is the source of nutrients for those cells. So you have to keep replenishing media with all of the growth factors, all of the nutrients, and therefore the main, and additionally also for the cells to survive, because they are not surviving under normal circumstances. Therefore the pH of the medium in which the cells are present is also extremely crucial. So here the reason for using a pH indicator is therefore twofold. One is to make sure that the pH is fine. You make sure that the media, the culture medium in which the cells are being allowed to grow, that medium is it's not exhausted, it is not deprived of nutrients, because the if you have say 10 cells, and from 10 cells over a period of say 48 hours you are getting 1000 cells, then the same amount of media will not allow it to persist for an extended period of time, because those cells will essential factors like survival essential factors will utilize that to multiply. Now for these 1000 cells to survive, you need more media and you need to replenish it, and therefore a pH indicator is extremely crucial for such steps, because it indicates that the medium is no longer buffered, it has changed pH, it has become acidic. It means that the cells have produced metabolites, and therefore you need to provide them with fresh media. So after this elaborate discussion about the necessity of pH indicators, we are coming to the one which is most commonly used. So in the options, we have methyl red, phenol red, neutral red, and thymol blue. But among these, phenol red is the commonly used indicator for most mammalian cell culture media. There are exceptions to this, but RPMI and DMEM, which are among the most commonly used media for cell culture, mammalian cell culture. These are usually they compose or they consist of phenol red as the pH indicator for the purposes I have previously described. So this was also a direct question. Now, coming to the next question, it is asking us about the interface of the cell cycle and what does it comprise of. So first you need to understand that a cell cycle, um, what exactly is the interphase in a cell cycle? An interphase is the time difference or the time between two consecutive M phases. So any part of a cell's life where it is not actively dividing or not actively proliferating is actually part of the interphase. So if you have one division phase and another division phase, whatever phases are coming in between would be included under interphase. So by that definition and by the lecture notes that you have already received, if you look at the options, you will see G1S, G2, S, G2M, G2M, G1, and G1, G2, and M. So option B, C, and D, they include the M phase. But as we have discussed, this is the interphase, is the interfering time period between two M phases or two mitotic phases, and therefore we cannot have M phase as one of the constituents of the interphase. So the other options, which are G1, S, and G2, are actually what comprise the interphase of the cell cycle. So I will just take a 2-minute break. I need to drink some water and I'll be back. So coming to the next question, which was about which is the eighth question in this assignment. It was about the stage of mitosis where the nucleolus disappears. So among that, we have the options anaphase, metaphase, telophase, and prophase. So if you have carefully looked at the fourth lecture of this week, then which talked about the cell cycle and the details of each of these phases. So there are some key or some characteristic features, and among those we sorry, yeah, so those are the different phases that are present, and among those, prophase, which is the first subphase from the mitotic stages of a cell, or mitotic or M phase of a cell cycle. Prophase actually looks at or it has some characteristic features which include the disappearance of the nucleolus. So the entire nuclear envelope, it dissolves, and that is necessary because decondensation of the DNA, which has previously been present or has been compacted into an extremely condensed state, so that you can accommodate the huge amount of DNA which is present in the cell into a very small area, very small portion, and therefore the nuclear envelope that has to dissolve. The nucleolus, which is within the nucleus, that also has to dissolve, and then that decondensation can occur. The nucleus can double, and so on. The nucleus already has doubled, it can divide into two. So for that to happen in the prophase, the nucleolus disappearance actually takes place. If you look at the other options here, they have other characteristic features. Like after prophase, we have metaphase, where we know that the condensed chromosomes, previously the chromatin which was present, that forms these chromosomes, and during metaphase they all align along the metaphase plate, which is somewhere in the center of the dividing cell. And next, during anaphase, the two chromat, like if you consider these two as the sister chromatids, then the two sister chromatids are separated towards opposite poles, and that is done with the help of microtubules, which makes it a very characteristic feature of anaphase. And while these sister chromatids are being pulled back, like if you consider it like this, these are being pulled back, they take shapes like V, and usually these are characteristic features. So if you are asked to, or if you're provided a slide, like a microscopic slide, and you're just given a certain field to observe and identify what stage of the cell cycle is this particular cell in, then you need these characteristic features to actually identify the same. So that was anaphase, and in telophase, obviously you can expect that the cells have divided. They are completely towards opposite poles. They're very close to dividing, and during cytokinesis, they will ultimately form two daughter cells. So some of these are the characteristic features that we have talked about. Then comes the question of, like I was previously saying, that you need to have some characteristic features about each of the cell cycle phases and subphases on your mind. So here we've been asked about the characteristic feature of metaphase. So if you look here, the options are that A, the chromatin condenses into chromosomes. The yeah, the condensation process, as we have discussed, that occurs during prophase. The second is that proteins attached to each chromatid are cleaved and sister chromatids are separated as daughter chromosomes. Now that also we discussed that the migration towards opposite poles, that entire step, it takes place during anaphase. Then the chromosomes get aligned at the equatorial plane, so that occurs, that as we have discussed is obviously a feature of the of metaphase. And the fact that daughter chromosomes are moved and they attach to opposite ends of the cell, that occurs during telophase. So since the question is asked us, or is focusing mainly on the metaphase, the correct option is option C, where the chromosomes align at the or along the equatorial plane. So that covers this question. Then the next question was about lysosomes. Now lysosomes play a role in necroptosis, autophagy, apoptosis, and necrosis. Now necroptosis is a sort of a combined phenomenon where a cell is seen to it appears as if the cell is undergoing necrosis and apoptosis at the same time. There are clear distinctions between these two processes as well. Apoptosis is a much more regulated, programmed form of cell death, whereas under necrosis, a cell basically it swells up and it bursts and it dies. It's a unregulated, more a random, like it's a more it's not as fine-tuned a process as the other one is, so maybe you can look at it that way. However, lysosomes in particular are involved in this process called autophagy, where you can actually find these in the lecture notes as well, where Professor he has discussed about each of the organelles in separation. So there, autophagy, which is the process of chewing, where a cell basically digests itself. Like if a cell is deprived of nutrients, it does not have access to nutrients or it's not able to survive. Under those circumstances, what will happen is that it will basically consume its own components and it will just die. So that's what happens during autophagy. And lysosomes, because particularly because they are the key digestive components of any cell, they contain all of these enzymes and they are responsible for this process. Therefore, they play a role in autophagy. So that actually covers your question. Then we have been asked in question 11 about which of the genes are actually considered to be the guardians of the genome. Now, this is actually a very commonly used terminology for one particular gene, which obviously, as you can see in the answer, is P53. So what do you mean and why this name? Why is it called the guardian of the genome? The reason for this is that the entire process of cell division and particularly of genome maintenance, and like there is a terminology which is fidelity with which a process occurs, which means the accuracy, maintaining that if a particular region of the genome has ATGC sequence, then in its daughter copy or in the daughter cell, the exact same sequence should be copied without any errors. So that process, that accuracy is the maintenance of this accuracy is actually known as the fidelity. Now, that is just about replication, right? In general, during the process of cell cycle, because particularly when you are talking about meiosis, the process of meiosis, which is actually what occurs in the germ cells, where an individual's entire genetic information is stored and it has to be carried over to the next generation. So that is a very crucial process and that determines the entire survival of the offspring or of that child, of that individual. Therefore, this process requires very high fidelity, and at each point of the cell cycle, each um, junction of the cell cycle, there is a checkpoint, which has also been mentioned in the lecture as well. Checkpoint basically means that before you proceed from stage 1 to stage 2, before you can enter stage 2, the cell actually makes sure it checks, it verifies that yes, everything that is required for moving to stage 2 that is present, and these things have, it has a checklist and it makes sure that all of this is done. And one of the major proteins that play, there are other proteins as well, but one of the major group of proteins are the P53 groups of proteins which are produced by the P53 gene, and it is extremely relevant, it is extremely crucial in maintaining this fidelity, and therefore one of the most common mutations for almost all cancers. We cannot generalize and say this for all cancers, but for almost all of them, P53 mutation is a given. It is the major regulator of the fidelity via which a cell is allowed to replicate. If in case a cell has somehow incurred some sort of damage in its genome, then under normal circumstances, P53 will not allow that cell to divide. It will make sure that whatever damage has been incurred, that is getting repaired. And only after that repair, it will allow it, it acts as a traffic police, and it makes sure that, okay, if this is wrong, if there is an accident, this cell should not go further. It should not proceed for division. It's not fit to proceed for division. But when when crucial genes like this are undergoing mutations, then such cells, despite carrying such mutations, such very crucial mutations, or some sort of genomic, some sort of insult to the genome or some sort of mutation to the genome is present, and this, despite that, such a cell is still undergoing division and still proceeding forward, and therefore it leads to cancer, and they carry out uncontrolled cell division, and that leads to, of course, a large number of problems. Therefore, the p53 groups of genes, or particularly p53, is extremely crucial in acting as the guardian of the genome. Then in the sorry then in question number 12, we had been asked, after which phase of the cell cycle do these cells enter the quiescent phase? So the quiescent phase, if you remember, is the G0 phase. And if you recall the overall organization or the overall stages via which a cell undergoes division, then first we have G1, then we have S, then G2. G1, S, G2 together are part of interphase. Then the cell, after it has made sure that yes, all of these things are fine, G1 is the prep phase, S is the synthesis phase, G2 is the prep phase for the mitotic stage. Only after all these verifications have been done does a cell actually enter into the M phase or the mitotic phase. However, when a cell has undergone division and it has differentiated, which means that you started from a stem cell which has no particular specialized function or no particular morphology, shape, anything of that sort. It is not part of some sort of tissue. From that stage, when it undergoes differentiation and when it reaches this stage, it enters the quiescent phase, which is the G0 phase, which means that the cell is now quiet. The cell is not going to divide. It's not a growth phase. It's just a quiet phase. And one example of cells that have entered and that stay in the quiet phase are are neuron cells or neuronal cells or neurons. They are said to be end differentiated, which means that they remain in that quiescent phase. And this phase obviously comes after G1, which is where the cell has decided that no, it's not going to divide. It is going to stay in this phase because the cell has already differentiated, or it does not need to proliferate any further. So after G1, some of the cells can enter into G0, but under normal circumstances, if a cell is dividing, is healthy and is capable of dividing, and it has all of the growth factors and additional factors that are necessary for it to divide, then it will proceed towards G1 phase. So that covers question number 12. Then in question 13, we have been asked which of the following acts as an antigenic determinant in gram-positive bacteria. So I think we had discussed in the last session, but in case somebody was not there or you're not familiar with it, antigenic determinant basically means any protein can be considered to be, very broadly speaking, antigens, or any chemical entity can be considered to be an antigen if an antibody can bind to it, which means that if it is on the surface and if an antibody can bind to it, then that is an antigen. So in gram-positive bacteria, you can see here the cell walls of gram-positive and gram-negative bacteria taken from this website. So these are the two types of cell walls that are normally present in these two bacteria, based on which they are actually capable of taking up gram stain, and hence from there the nomenclature of gram-positive and gram-negative comes in. So here we can see that in gram-positive bacteria, they obviously have the cell membrane. They have a thick peptidoglycan layer, which has lipotecoic, teichoic acid, and some other surface proteins. Whereas if you look at the gram-negative bacteria, above their cell membrane, they have a thin peptidoglycan layer. It's not as dense or as thick. And then they have an outer membrane, a whole other structure. They have an outer membrane, and they have which is an LPS or lipopolysaccharide layer, and they have some different surface proteins in those cases. Now, since our question is asking about gram-positive bacteria and their antigenic determinants, you can see that among the options, lipopolysaccharides, eicosanoic acid, palmitic acid, and lipotecoic acid. The figure also shows, and by the lectures notes or the lectures that have already been discussed by Professor Trivedi, you can see that lipotecoic acid is the actual antigen that is present on the gram-positive bacterial cell wall, and therefore they are serving as recognition sites, or they are providing epitopes to which antibodies can bind, and they are therefore serving as the antigenic determinants. So this is the distinguishing character, one of the distinguishing characters between these two types of cell walls. Then we come to question 14. So this was a fairly direct question where we had been asked that porins in mitochondria, they allow molecules with a molecular weight of what? So porins are, as the name indicates, they are sort of pore proteins that provide a sort of pore or a channel through which certain molecules can be allowed to pass, and that deciding factor or that distinguishing factor based on which a molecule would or would not be allowed to pass is usually its molecular weight. And in this case, it's obviously a very direct question. If you look at the options, there are 20,000 Daltons, 50,000 Daltons, 5,000 Daltons, less than 5,000 Daltons, or more than that. So porins, because they sort of form a pore-like structure, they form a channel through which only a particular restricted size of proteins can pass. Therefore, the answer for this question was less than 5,000 Daltons. So if a protein is small enough where it is less than 5 kilodaltons, then that is allowed to pass through the mitochondria, and therefore it restricts what can or cannot enter the mitochondrial environment. So that was this question. And for this week's assignment, the final question was about ATP synthase. So ATP synthase, or F1FO ATPase, is a dash-shaped multimeric protein complex. So if you remember the structure, then it had an F1 region and F4 region. So altogether, it formed a mushroom structure. The other options, clover leaf structure, is usually associated with or is usually the terminology that is utilized for the structure of tRNA, which is obviously necessary for the process of translation. It brings charged amino acids, which can be incorporated into the growing or synthesizing peptide, and the final protein can be formed. We have these in the next week's topic as well. So that is the clover leaf structure, and L and V shaped are usually associated with the chromosomal structures that are seen during the process of anaphase when sister chromatids are being taken to the opposite poles. So none of these are the correct options here, obviously, and the appropriate option for this case is the mushroom shaped multimeric protein complex. So that's what we see here. So with this, we come to an end for this week's questions. For some reason, these questions are, I don't know why those questions were not visible, but next we'll discuss some sample problems for the upcoming week's lecture, so that even though you might have submitted some assignment, we just are discussing a few more sample problems. We can discuss week 4 assignments in the next week, and I'll just be back after a 2-minute break. Okay, so there's some issue with the animation, so I'm just going to present it this way, and I think it would be visible. So, some sample problems for week 3. Based on the topics that are covered, here are some questions. So, the first one is actually talking about, let me try to remove this. Yeah. So, the first one is talking about the only component in a nucleotide that has a net negative charge at physiological pH. So, you have to first keep the components of a nucleotide in your mind. So you have what? You have a nitrogen base, you have a phosphate, and what the question is asking for is the negative charge. So at physiological pH here is obviously it's relevant because the pH will actually determine what is the charge on any molecule. But under normal circumstances within a cell at physiological pH of 7 to 7.2, the only component of a nucleotide that will contain a negative charge is the phosphate, the PO4 2 minus area, or the phosphate basically region will have that negative charge, and that is actually the component which confers a negative charge to DNA as well. That affects its migration through an agarose gel, as we have discussed initially during this interaction session. So that gives you the answer to this question. The next question is about the major force that stabilizes our double-stranded DNA in an aqueous solution is. So dsDNA, so the reason why DNA, as you are familiar with it, has that double-stranded structure is because there are multiple forces which stabilize it. If you look at the options here, there is base stacking, hydrogen bonding, hydrophilic interactions, and ionic interactions. So while every interaction has some role or the other to play, among these, the major contributing force that actually stabilizes this structure is the base stacking interaction. So if you have two strands of DNA, one strand has A and that is forming a double bond with T at a particular location. And after that A, if you have another T while the other strand has an A, then these AT and TA bonds together are stacking against each other. They are forming pi-pi interactions. These base stacking interactions are providing additional stability. So therefore, the formation of one base pair is accelerating or is cooperatively supporting the formation of the next base pair, and this keeps on continuing until you have the entire double-stranded DNA structure. So that is what will stabilize the dsDNA structure in an aqueous solution, and therefore among all of the other options, the base stacking interactions have the most crucial role to play under these circumstances. So that was question number 2. Then the third question talks about the type of isomerism that occurs in nitrogenous bases that involves a shift in the position of hydrogen associated with either nitrogen or oxygen. Here we have skeletal isomerism, geometrical isomerism, tautomerism, and conformational isomerism. So this is also a fairly direct question. If you've gone through the lectures of week 4, I'm sure you have. The assignment submission is done as well. But obviously tautomerism is the answer to this. It's a fairly direct question. I don't think there's much to explain here. So that's pretty much it. Then comes the Shargav's rule. So if you remember, we were talking about base pairing just now. So Shargav's rule is not applicable to which of the following? So Shargav's rule, first let's discuss what is it. So Shargav's rule actually states that A and T will form two hydrogen bonds with each other or will double bond with each other, and G and C, they will form three bonds with each other, or triple bond is formed between those two nucleotides. So that is Shargav's rule. So as you can understand, that is actually what supports the idea of complementarity among the two strands of DNA, and that's actually what is the major driving force. So if you look at the other options here, if you look at all of the options here in general, you have dsDNA, dsRNA, RNA-DNA hybrid, and ssDNA. So because Shargav's rule is talking about two nucleotides or two bases which are able to form bonds with each other, therefore a single strand or a single nucleotide polynucleotide strand will not actually follow this rule because it always needs a complementary region, or it needs a complementary base to actually execute this um, to actually follow this rule, and therefore ssDNA is not the answer regarding this. Then comes and sorry, ssDNA is the answer. It's not actually following the Chargaff's rule. Sorry, apologies for the mistake. Then comes question 5, where we have talked about, or where the question actually talks about the Sanger's method. And in Sanger's method, the step for chain elongation, it stops DDNDPs, which is dideoxynucleotide triphosphates, due to what reason? So if you go through Sanger's method, it's also known as the dideoxy method. And the reason why it is the process of chain elongation stops or chain termination occurs is because instead of a DNTP, a DDNTP gets incorporated and chain elongation can no longer continue. Now the process of chain elongation is actually the formation of phosphodiester bond, and that bond is formed between two specific atoms, the two specific groups, the 3' OH and the 5' phosphate. Therefore, because a dideoxy NTP, it lacks the 3' OH group, therefore extension is not possible, and therefore the answer to this question, as you can also see from the tick mark given beside it, is that the 3' OH group, so which is absent in the dideoxy NTP, is the reason why chain elongation cannot continue. And it's actually the reason, and because for each reaction mixture both re-NTPs and DD-NTPs are provided, therefore you expect that in every reaction setup at different positions the DD-NTPs would get incorporated and would give you extended polynucleotides of different lengths, and based on that you can actually trace it back to identify where exactly one particular nucleotide is present. So for example, if you're looking at the reaction setup for DD-ATP, then you would understand where exactly A's are located, and so similarly for all the other nucleotides as well. Then in question 6, we have been asked, which is the most dynamic RNA that undergoes continuous turnover and it acts as a template for translation? So even if you're unsure about the first part, which is possible because there are several different types of RNA which are present inside the cellular soup, you can still look at the second part of the question where you have been asked that the RNA that acts as a template for translation. So if you remember the central dogma clearly, then you have the entire genome, the DNA, from which you produce, from which transcription is actually occurring, and it is decoding the information that is placed within the genome, and it is producing mRNA or messenger RNA. Why messenger? Because it is carrying the message present in the DNA and it's allowing its conversion into proteins, which are formed in the third step of the central dogma by the process known as translation. So if you look at the options given here, we have guide RNA, mRNA, miRNA, and tRNA. Now, guide RNA is a part of the CRISPR-Cas protocol where, or it's also involved in siRNA-mediated silencing. So the guide RNA is, as you can understand, it acts as a guide, right? It acts as a guide for something else, for some process to take place. Majorly, it is for silencing or um, knockout or removal of some region. Therefore, they do not act as templates for translation. And they, while they do um, undergo turnover, it's not as rapid as that of the next option, which is mRNA. So mRNA obviously acts as the template, and it obviously also undergoes quite rapid turnover. They do not have very high stability, and the reason why this is actually essential for the cell, because it might come up as a question that, you know, if you are spending so much energy into transcribing some gene and you're producing its mRNA, then why are you not allowing it to persist indefinitely? The reason for that is that any gene, its expression, that constitutive expression is only allowed for very selected, very limited number of genes, while most genes are actually placed under spatio-temporal control that only in a particular cell and only under certain circumstances should a gene be expressed. And that control is necessary for overall survival of the cell, to conserve the cell's energy and to also support its survival, because not every process is necessary at every point of time. So therefore, that is why RNA turnover is also extremely important, and it is regulated very finely as well. So that is what we were talking about mRNAs. Then came miRNA, microRNA. They also have a different function that we've already read about. They are part of the silencing process as well. And finally, tRNA, as we have previously discussed, they have that clover leaf structure, and they are involved in translation, but they definitely do not act as as a template for that process. They are actually involved in a more complex and more finely regulated step, which is to bring the correct cognate, like tRNAs have to undergo charging, and by charging we mean that the tRNA anticodon loop, which is used to recognize the correct amino acid sequence, or rather you can say the codon from the mRNA, that is recognized by the anticodon loop, and the cognately or the appropriately charged amino acid is incorporated into the currently synthesizing peptide. So it performs a different function. It does not act as a template, but it is involved in translation. So you need to keep this in mind when you're answering questions like this, where the options are really close by, but one of them is definitely not accurate. So it just needs a little more uh, time that needs to be given, and more accurate reading of the questions that you've been provided. Uh, yeah. Then in question number 7, we are being asked the nitrogen containing structural polysaccharide that is found in the cell walls of fungi and in the exoskeleton of arthropods is A pectin, B chitin, C cellulose, or D hemicellulose. So whenever we think about cell walls, we directly go towards cellulose, but this is a very common mistake that is made by a lot of students and should be very accurately studied. It is that only plant cell walls are actually majorly composing of cellulose, but arthropods, or basically insect exoskeletons, the outer hard covering or shell-like structure which is present for most insects, and also in this case, as has been mentioned also in case of fungi, they contain of a different component or a different structural polysaccharide, which in this case is chitin. So chitinous exoskeleton or chitinous cell wall is a characteristic of arthropods and fungi respectively, and therefore the answer to this question is obviously chitin. So that completes this question. Then we are moving on to the next, which is also quite related to the previous one. So the repeating units of cellulose are called as A, hemicellulose, B, trehalose, C, cellobiose, or D, sucrose. So cellulose is actually made up of, so this is a fairly direct question. If you go through the lecture notes, you'll see that these are polysaccharides, which are obviously composed of monosaccharides. So the monosaccharide units that actually comprise the cell wall of plants in this case are cellobiose. So cellobiose repeating units together form the polysaccharide entity, which is called as cellulose, which composes the cell wall of all plants, and yeah, and therefore the enzyme, that is only enzyme which is able to break it down, is cellulase. And because humans, we don't contain cellulase, we are not able to digest grass and other plant products directly, which are very cellulose rich, and yeah, that is the distinguishing criteria. Then comes the end product of protein catabolism in humans. So again, a very direct question. The options here are urea, uric acid, glutamine, and ammonia. So upon complete, so catabolism of course means the breakdown. So when proteins are completely broken down in human beings, it ultimately leads to the formation of urea. So this also you will also find in the lecture notes, and it's extensively discussed in detail. We will also pick it up in the next week's session on Monday, where we'll take up week 4 assignments and questions, and also the previously discussed topics. And therefore the end product for this process is the production of urea. And in the last sample question, we have Krebs cycle. We have actually discussed this before, but yeah, it's pretty much related. It's one of the reasons why you should go through both. You should try to keep up with the assignment, like the lecture videos for at least the next week, because many a time a lot of questions are quite interrelated, because eventually so is biology, right? So we were talking about Krebs cycle previously. So this question is asking about where it takes place. So the options here are mitochondrial stroma, cytoplasm, lysosomes, and mitochondrial outer membrane. So the option, of course, is mitochondrial stroma, and that pretty much covers our sample problems and our assignment that has already been submitted for week 3. So next, we will try to recap some of the topics based on what has already been discussed in the lecture notes. We'll try to summarize all of the things and all of the topics that Sir had previously discussed, and we'll then proceed forward with it. So after just a minute's break. Okay, so coming back to the discussion, we are going to recap most of the topics that had been covered during the lecture by Professor Vishal Trivedi. So in the first lecture, he discussed about the diversity in living organisms in general. And as we have already previously discussed, we have talked about classification, about evolution, and now we are trying to understand cells. And in the upcoming weeks, more of these topics have been taken up. You might already be familiar with a lot of them as well. So he talked about how he explained how cells can be understood based or cells can be studied based on their shape, size, spatial distribution, and biological activity. But then when we talk about cells, what are they? Cells in general are considered to be the structural and the functional unit of life. So if you take up each of these terminologies individually, then it means that when first let us try to understand why is it the structural unit of life. The reason for that is that the structure, it is the okay, so if you think about just a molecule is made up of atoms. So that is the structural unit which makes up an atom. However, if you talk about an entire cell, then the structural unit of life or of a living organism, one cell is giving rise to tissues. That tissue is giving rise to organs. The organs are giving rise to organ systems and ultimately the organism. So therefore, it can be considered to be the structural or the basic unit of life. You talk about structure. On the other hand, the functional unit of life means that for a living organism, all of the basic processes which are necessary for its survival, which includes its ability to, to breathe, to respire, to metabolize, to survive overall, all of the individual digestion, excretion, respiration, all of these processes, these are carried out by either an organ or an organ system, right? So as I just mentioned that these organisms are fundamentally made up of one type of cell, right? Or a group of cells, they're made up of cells. So individual function performed by each cell is actually helping the entire organism survive as a whole. So cells are considered the structural and the functional unit of life. So that is the most commonly used but the most relevant definition of what a cell is. So after that, we are talking about how cells can be classified as prokaryotic or eukaryotic. So here you can see that a prokaryotic cell, most common example is a bacteria. They are single cells, whereas eukaryotes are, they are also capable of being single cells. Like there are unicellular eukaryotes which are present. Example being the entire kingdom of protozoa, as we have previously discussed. But apart from that, there are majorly eukaryotic cells. A large proportion of them are actually multicellular in nature, and of course, plants and animals are the most common examples of eukaryotes. So if you talk about the major distinguishing characters, then we have all of these. So first is the size. Prokaryotes are obviously very small in size. They are few very small micrometers in their dimension compared to eukaryotes, where they can go up to around 40 micrometers in their diameter. They are much larger in comparison. Then comes the genetic material. So in case of prokaryotes, their genetic material is not, they don't really have a well-defined nucleus per se. They usually have a nucleoid or an ill-defined sort of structure, which is here, as you can see, it's circular DNA that is present in the form of a nucleoid or as a free material. Diagrammatically also you can see it here present. Whereas eukaryotes have well-defined double membrane nucleus that is very well separated from the cytosol, and that compartmentalization is necessary for some of the most crucial life processes of eukaryotes to actually take place. And within that well-defined double membrane nucleus is the linear chromosome, which is the genetic material for eukaryotic cells. So that is how they're different as well. Then the third property is in terms of replication. So prokaryotes replicate from a single origin or one ori sequence is present, whereas eukaryotes have multiple origins of replication via which it takes place. Then come the genes. So when we talk about genes, there are, in case of prokaryotes, there are no introns, whereas eukaryotes obviously do have introns. They have interrupted genes, which are interrupted by introns in case of eukaryotes, whereas prokaryotes do not have those structures. Then come the organelles. So organelles are not membrane bound, whereas eukaryotes are membrane bound. Cell walls, prokaryotes have complex cell walls present, but eukaryotes do not, except of course fungi and plants. animals do not possess a thick cell wall. The ribosome is also different in terms of its sedimentation coefficient, which is 70s and 80s respectively, and also the process of transcription and translation do not occur in separated compartments for prokaryotes. They occur co-transcriptional translation is what takes place in prokaryotes, whereas in eukaryotes they are separated in the nuclear and cytosolic compartments exclusively. So these are the very fundamental differences between these two. So obviously you can see that prokaryotes are simpler, smaller, less complicated. You have the nucleoid DNA, you have plasma membrane, cell wall, the slimy capsule layer, which is something we've also talked about previously. Then ribosomes are there just lying around in the cytosol. There is no distinction between a nucleus and a cytosol. They have pili, which are little locomotory structures which are necessary for those cells to move. This is a plasmid DNA. So you can have both nucleoid DNA, which is like unorganized chromatin material, and you can also have extra chromosomal DNA, which is plasmid DNA. And here you have mesosomes. Mesosomes are basically infoldings of the plasma membrane. Um, yeah, infoldings or inner invaginations of the plasma membrane, which are present in prokaryotes per se. Then we talk about the outer flagella, the flagellum attached to the bacterial capsule. This is what you see here. It is majorly responsible for one crucial function, which is chemotaxis. It's necessary for locomotion, for movement of the bacterial cell, right? They do not have other appendages. So they need to be propelled through whatever surface they are moving through, whatever medium they are moving through. And for that, this flagellar structure is extremely crucial. So additionally, the flagella structure, if you go into it, it's quite complicated and it's a very energy expensive process. So it's made up of flagellin, and this flagellar protein is actually the reduction or suppression of this actually affects its pathogenicity and its ability to grow. So this is the flagella that we talked about. Then come the surface layers. So bacteria, we will discuss this. So bacteria possess three anatomical layers. The first or the most external or outermost layer is the bacterial capsule, which is made up of polysaccharides. It's made up of sugars and polysaccharides, and it's also like a tough covering which is present on the bacterial cell. And one of the reasons why this tough covering is a characteristic of bacterial cells is because they need to survive under very adverse conditions in very rough and not so comfortable environmental as well as predatory conditions, and therefore they need to have a tough coating which can help them endure all of the changes and can help them to adapt under very extreme environments as well, which brings us back to the extremophile discussion that we've previously had. It's impermeable to the what? Of course, extremophiles have other contributing structures or proteins as well, which help in their survival. But yeah, capsule is a general component of most bacterial cells, and it's impermeable to water, other aqueous solvents. It has some proteins which contribute to the antigenicity of the bacterial cells, and a cell wall in bacteria and its response to gram staining is one of the ways in which bacterial species are actually classified. So that's another very relevant contributing factor. Then comes the structure of the prokaryotic wall. We have discussed it as well, gram positive versus gram negative. So peptidoglycan is the most not relevant, the most visible distinction between these two. So it's made up of NAG and NAM residues, an acetyl glucosamine and an acetyl muramic acid, which are linked by beta-1,4 glycosidic linkage. And the other amino acids which are present are L-alanine, D-glutamic acid, L-lysine, and D-alanine as well. They are cross-linked, they form a meshwork, and they provide the physical rigidity of the cell wall, which is one of its key functions. So penicillin-like antibiotics actually target the synthesis of this peptidoglycan, whereas lysozyme, which is present in human tears and saliva, it's actually capable of cleaving this glycosidic bond between NAG and NAM residues, and therefore it's able to harm the bacteria and it's able to protect against bacterial infection. That's why you have lysozyme in the tears, right? So that you don't catch eye infections very often, because that would be difficult to tackle under other circumstances. So that is there. Then yeah, sorry. Then we also have the lipoteichoic acids, which we also discussed in the assignment. It's an important antigenic determinant. It's only found in gram-positive cell walls, bacterial cell walls. Then lipopolysaccharides on the other hand. So you have to at least try and avoid confusion between these two terminologies. So lipoteichoic acid is in gram-positive cell wall, whereas lipopolysaccharides are a characteristic feature of gram-negative, and in those cases they act as the antigenic determinant. And the explanation for gram staining was also given here with crystal violet and safranin. Then we come to cytosol and other organelles. So prokaryotic cells do not have any membrane-bound organelles. They have a 70S ribosome, genetic material, and the ETC complexes which are necessary for generating or producing energy. They are also present there. Then the chromosome and the extra chromosomal DNA, it's also there. The bacterial chromosome or circular DNA, the plasmids that are present. So that's actually the genetic content of a bacterial cell. Sorry. So the different forms of plasmids, bringing us back to the question, we have the relaxed covalently closed circular DNA, which can be supercoiled by DNA gyrase, which can be cleaved by endonuclease to form open circular DNA, and can be attached by DNA ligase to form this covalently closed circular structure. This can be also nicked by endonuclease, and the supercoiled one can be resolved into topoisomerase. So this is the relate or interconversion from one form of the plasmid to another can be carried out by these enzymes. So these are the different forms, and we have talked about how their conformational differences also affect their electrophoretic mobility or their ability to migrate through an agarose gel. When the bacterial plasmids, what are their immediate components? So we had the origin of replication, the antibiotic resistance gene, and majorly these two are required for any plasmid to be able to sustain over multiple rounds of divisions. It needs to be able to replicate, and that is under the control of the origin of replication. Whereas what actually gives that bacterial cell an advantage or an edge, or what makes it very relevant for our use in terms of recombinant DNA technology, is the presence of an antibiotic. It is the presence of a selectable marker, which is majorly an antibiotic resistance genes, and that gives it some advantage over the other bacterial cells. So that's a bacterial plasmid. Then here we have the protocol for its isolation as well. The three different solutions, where we first have the resuspension buffer. It's the alkali lysis method. So first is the resuspension buffer, where we have glucose, tris hydrochloride, and EDTA. Then we have solution 2, which is for lysis, which is SDS and NaOH. Then the renaturation step, where only the plasmid DNA would be able to, the neutralization step or renaturation step, only the plasmid DNA is able to recover or is able to renature because of its smaller size, but the much larger chromosomal or genomic DNA is not able to do so. That's how you actually eliminate genomic DNA contamination. And also, to avoid any protein contamination, or at least to try and avoid it, we go for PCI method. This was one of the sample problems we discussed also in the last class. And phenol chloroform isoamyl alcohol or PCI mediated extraction is carried out to remove any proteins in the precipitate. And finally, we can precipitate the plasmid by 100% ethanol from the solution. So this is how the migration actually varies, and this shows what we have also discussed in the question. So then this was a summary for the same. Then we'll come to the second lecture. Yeah, so it's loading. Yeah. So after the discussion on prokaryotic cells, next we in the second lecture we had moved on to eukaryotic cells, and there we find very distinct and very different characteristics between plant and animal cells. So while plant cells majorly have a cell wall, animal cells, in case of animal cells, those are absent. Plant cells are quite large in size, animal cells are much smaller. Then the presence of chlorophyll, obviously it's not present in animal cells, but plants do have them. Vacuole, a large central vacuole is found in plant cells, but in animal cells it is absent. Sorry, not absent, it's much smaller, and they are like in case of plant cells, the vacuole has a whole other function in terms of turgidity of that particular cell, whereas for animal cells there are tiny vacuoles which are multiple in number. Then come mitochondria, which are few, but in animal cells they are much more. Lysosomes, they are almost absent, but here they are present. Glyoxysomes, present and absent respectively. And cytokinesis occurs by different methods, by plate and by constriction method. So these were the differences. Then he discussed about what is the cytosol, what is its function. It shows that salt to gel transition, the difference between cytosol and cytoplasm, where cytoplasm consists of cytosol plus organelles, but cytosol only consists of the liquid part of the cell. So there's a difference in the terminology, although they are quite often used interchangeably. The definitions for the two are actually different, and therefore we should try and maintain that distinction between the two. Then comes the nucleus, which is the central processing unit of the cell, and inside is the nucleoplasm. Here we see a cross section as well, the nuclear envelope, the chromatin inside, the nucleoplasm, nucleolus, and nuclear pore. Majorly its function, it has all of the enzymes and the nucleotides that are necessary for replication, transcription, DNA damage repair, etc. It shows the condensation of the genetic material into nuclear bodies or into chromosomes, and it's actually the chromatin can be divided into euchromatin and heterochromatin, and it's actually responsible for controlling which regions of the genome would be expressed at one particular moment and which would not. So that's the distinction. Then comes the nucleus. So nucleus in eukaryotic cells have the nuclear envelope. I mean, continuing with the nucleus, we have the nuclear envelope. The outer membrane is actually continuing with the endoplasmic reticulum. Therefore, you'll always find a continuous network. So if you have the outer nucleus, in continuation, we have this sort of membranous network, which is the ER. And the space between these two membranes is the perinuclear space, and the nuclear envelope often has little pores, and that's around estimated to be around 3,000 to 4,000 pores per envelope. Its dimensions and its functions were also discussed, and from there we move on to the mitochondria. So commonly we know that it's the powerhouse of the cell, actively involved in ATP generation, and it has porin proteins which allow less than 5,000 Dalton proteins to actually pass through, which is something we discussed as well. And mitochondrial targeting sequences can also be seen. So if a protein is destined to be sent to the mitochondria, then it will have this MTS or mitochondrial targeting sequence at the beginning, and only based on this address will it be redirected to the correct position. So that is what we talk about here. Then the inner membrane has these cristae that are present, and they occupy a large portion. It's actually allowing a much larger surface area to be accommodated within a much smaller space, and that has the machinery for anaerobic oxidation and the electron transport chain. And also the other substructures that we talked about, like the intermembrane space and the matrix, are also present. And apart from that, the intermembrane space, proteins present in the IM space, are responsible for apoptosis, or the process commonly known as programmed cell death. And matrix actually contains the ribosome, RNA, DNA, and the enzymes for Krebs cycle to actually be carried out, which was also the assignment question. Mitochondrial DNA or mtDNA is circular. It has its own RNA, mRNA, rRNA, and tRNA, and therefore there is a distinction between the normal nuclear DNA and the mitochondrial DNA. Mitochondrial DNA is inherited differently. Majorly, there is maternal inheritance, that is from the ovum. Directly the mitochondrial DNA is inherited into the zygote, and its paternal mitochondria does not really contribute to the same. So that we see. And then we see the entire process of electron transport chain. I'll not get into details of this because it has been extensively discussed by Sir. So here you'll find the F1 FOATP is the mushroom shape structure that we talked about. So the entire process of energy production is carried out in the mitochondria. So that is its major function. Then there is generation of reactive oxygen species. It is because there is maternal inheritance, you can actually track a family, and it has a major role to play in apoptosis as well. So apoptosis is also defined here because it helps in releasing cytochrome C. That's an important part. And here you see the process of ATP synthesis explained very well via the chemiosmotic hypothesis. And it was also shown via a very excellent video. So you can refer to that one more time if you need it as well. Then coming to the third lecture, just let it load. Yeah. So in the third lecture, we have looked at the chloroplast, the next organelle. There, where are they present? Plants, algae, other lower invertebrates, etc. Its utility, its location within the presence of the chloroplast. Within the chloroplast, where exactly is chlorophyll present, that has also been mentioned in the thylakoid. Then the different proteins like TOC, TIC were mentioned. The different subcompartments within the chloroplast that were discussed extensively. Then here you can see where exactly these photosynthetic machinery are present and how these thylakoids are actually stacked one on top of the other to form the grana or the granum. And the thylakoid membrane contains this green colored pigment chlorophyll, which addresses one of the questions that were asked in the assignment as well. So then we talked about the Z scheme. You can see it here as well. It's not as prominent, but yeah, the basic process via which the absorption of photons and the breakdown of water is ultimately leading to energy equivalence being produced is via the Z scheme. So there you can see the PS1 and PS2, which are playing major roles here, how they absorb photons and how water breakdown of water or catalyzing the process of water breakdown is ultimately leading to the structure. And you get ATP and NADPH, and together these energy equivalents are used for the Calvin cycle, which assimilates the CO2 present in the environment to ultimately form the plant food, which are basically sugars like glucose. So that was there. Then we came to the organelles that are involved in an entirely different function, which is vesicular trafficking. Majorly, first we talked about how there is a process of endocytosis. You are taking in something inside the cell, which is the process of taking up solid material from outside to the interior of the cell, that is endocytosis. Whereas when you take up water, that water or other liquids, that is pinocytosis. And similarly, when you secrete something outside the cell, that process is known as exocytosis. So that's what we initially discussed. Then endosomes are the basic membranous structures via which you can traffic proteins and other cellular components from one part to the other via this vesicular trafficking process. So you can see how after for example, let's take a plasma membrane protein. So after it has been synthesized by the ribosome, which is attached to the endoplasmic reticulum, or it is synthesized at the RER, it goes through the cis-medial and trans Golgi via endosomes, and it is ultimately allowed to be, or it is ultimately deposited or positioned at the cell exterior or at the cell membrane. So that's what we see there. Then yeah, then this is the ER, which has been expanded in significant detail. The rough endoplasmic reticulum and the smooth endoplasmic reticulum, both of which have been shown here, and how they differ, what are their functions, like steroid hormone synthesis, detoxification, sequestering calcium. In fact, the sequestration of calcium within the ER is a major contributor towards maintaining so calcium is a second messenger within the cell. It's actually responsible, the levels of calcium within a cell are responsible for some signaling events. It's taken as a signal that, okay, something is happening. So under normal circumstances, the ER acts as a storehouse for this calcium and does not allow its untimely release, or it actually controls its release quite stringently. So that is one of its major functions. Then of course the synthesis of proteins, phospholipids and carbs. It's sorting to different organelles and other modifications which have not been covered here, such as glycosylation. After the ER comes Golgi, and Golgi was visualized by Camillo Golgi first, hence the name. Then the different phases of the Golgi, the cis, medial and trans Golgi, and how it ultimately gets sorted at the predefined destination. So that we see there. Then come the lysosomes, which also we have discussed. They sort of are known as the suicidal bags because they have a very unexpected role to play in terms of autophagy, which is which is a sort of survival mechanism for cells when they are extremely nutrient deprived and they need to survive under those circumstances. So they degrade the ingested food material, they degrade pathogenic bacteria, degrade old proteins, and they are basically the suicide bags. Then we come to plasma membrane, which is commonly underestimated as just an outer covering, but no, it has a lot of functions which have been discussed by Sir as well, and that includes its act as a barrier, as a semi-permeable membrane which selectively allows some things to enter and some to not. It has several proteins which control the influx and efflux of other components as well. It maintains a difference in the environment. It maintains an, or you can say it maintains an electrochemical gradient, maintains the sanctity between the cell interior, cell exterior in terms of pH, in terms of ionic concentration, in terms of many other things. So it has several other roles to play. It performs as a scaffold. It is able to detect when a cell is not healthy and it needs to not detect, it's able to signal that okay, a cell is not healthy, it needs to undergo some sort of process and it needs to die. So all those major decisions are also a responsibility of the plasma membrane of the cell. So that is the scope till which the third lecture was discussed. And finally, in the fourth lecture, the major topic that was taken up was actually the cell growth, cell division. So here you see first he actually focused on the bigger components, the smaller components, and what are the building blocks of those smaller components. So like proteins, amino acids, and the building blocks are majorly these atoms. Then carbohydrates made of sugar, lipids made of fatty acids, and DNA and RNA made up of nucleotides. So those are the composing factors. So here we look at the mammalian cell culture medium, example being DMEM. So in DMEM, the pH indicator, as we have previously discussed, is obviously phenol red. And apart from that, you can see that except for the media composition, which is now available in like packed, commercially available in the packed format. Apart from that, you need to add sodium bicarbonate as a buffer, FBS for all of the growth factors, fetal bovine serum for all of the growth factors that are necessary for cells to survive, particularly if you talk about cancer cells. And an antibiotic like penicillin, streptomycin, this is the most commonly one used. We use penstrep in our labs as well. So penstrep is used to prevent any sort of bacterial growth, any contamination basically. So because mammalian cell culture, the sterility, the extent of sterility that is required for mammalian cell culture is extensive, and any bacterial contamination is it's something we cannot afford. So therefore, a lot of care has to be taken in that regard as well. So that's what we saw here. Then the cell cycle, the most relevant topic in this case, I think. Sorry. So the cell cycle, majorly, as you've already understood, it's the process by which the entire life cycle of a cell, and it's also cell division is the process by which one cell is eventually producing daughter cells or progeny cells. So there are majorly three phases: the interphase, where you have the synthesis of genomic DNA and cytoplasm; the mitotic phase, where you are dividing that genetic material into two halves; and then the cytokinesis, where the entire cell is producing two daughter cells. So first interphase, it's the prep phase, and it has the G1S and G2 phase, as we have addressed in the assignment as well. So if you look here, this is the M phase. So from the start of one M phase to the start of the next M phase, the entire this region is called as the interphase, as is shown in gray. This one, right? So after M phase, first you have the G1 phase as part of interphase. It is the growth phase, right? It's from the end of mitosis that mitosis is complete. One cell is divided into two, and what is happening here is that cellular proteins, enzymes, etc., all of that is getting synthesized. What is required for DNA synthesis in the next round, that is also synthesized during this phase, and it is very hugely under the control of p53 gene products, which also, as we have discussed, are considered to be the guardians of the genome, because they are operating at these very crucial checkpoints and ensuring that a cell should only divide when it is healthy, when its genetic material and overall the cell by itself is healthy. So that's a very, very important contributing factor. Then comes the S phase. So after G1 phase comes here the S phase, which is S is for synthesis. And during this phase, DNA synthesis is carried out, and all of the chromosomes in the which are present in the nuclei, they are replicated. DNA content is doubled. There's no change in ploidy. Here synthesis occurs very fast. So because you do not want, because the newly synthesized DNA is not really protected, and it could be quite prone to mutations, and you don't want that. So process of synthesis is very, very fast, and it has to be done with a huge preference for not preference, it's actually a huge fidelity requirement is there, that whatever is getting replicated should get replicated correctly. So that occurs during the S phase. Then the G2 phase is the second growth phase, where the proteins that are required for mitosis and cytokinesis are synthesized. Then the G0 or quiescent phase, which is also something we talked about, it occurs after the G1 phase. So after G1, instead of entering S phase, there's another alternative, which is for the cells to get redirected towards the G0 phase, and never will a proliferating, fast-growing cell will never enter into G0 phase. It's not a regular condition. Only under specific circumstances will it do so. Sorry. Then comes the cell cycle. So mitosis or M phase. After the G2 phase, cells enter into the M phase, and there the entire process of DNA division takes place. Here you can see actually the varied extent to which a cell is dividing, and you can see that it's not necessary that all of the cells will be synchronized will be at the same stage of division, and therefore you can see the huge variety that is there. And under M phase, each of the different subphases were also discussed. Like we've previously talked about some characteristic features of each phase, such as the prophase, the metaphase, anaphase, and of course the telophase. So after all of this is done, where within a dividing cell at the two poles or at the two extremes, you can see the genetic material has separated. After that is the process of cytokinesis, where cell division, it requires distribution of the remaining cellular content equally, and for animal cells at least a cleavage furrow is formed, and individual nuclei are also able to divide, and all of the cell organelles also should be divided, and this should also be done with a lot of accuracy so that equal division of all components takes place. Now, one of the most basic questions, why is cell cycle at all required? So it's required for development and growth. It is required for replacing previous cells, because no cell can proliferate indefinitely. Then it is required for regeneration as well, for asexual reproduction as well. And then the control of the cell cycle is via those cell cycle checkpoints, which we also have previously mentioned, which are necessary to ensure that completion of different steps is occurring with a lot of fidelity, and in case there is any DNA damage, that is getting repaired. And there are three major checkpoints which are present, which are G1S, G2M, and the M phase checkpoint, and these are actually also taken care of by the cyclin. It's actually the transition from one phase to another, which is executed by cyclin and cyclin-dependent protein kinases, and the p53 gene products, which altogether regulate each of these events. And if under any circumstances these are not carried out with fidelity, or if there is something that is going wrong under these circumstances, then such cells give rise to cancer. They are cancerous cells if they are not obeying these normal rules of cell division, and therefore they will indefinitely proliferate and they will give rise to tumors, and that's obviously not something that's desirable. So that is what we have covered today, and uh, since I don't think there are any more questions, so what we can do for now is we can end the meeting, I think. And if there are any questions, you can post about them in the discussion forum, and we can address those in upcoming sessions. And in the next session, live interaction session, what we will do is we will cover the syllabus for week 4. We will look at the assignment that you've already submitted, and we'll also try to look at some sample problems for the next assignment to help you address those questions. So with that, I will conclude today's session. Thank you so much.